Welcome to the review session for Mendelian exceptions and extensions. Question 1. If four sisters all have different blood types, A, B, AB, and O, what had to be the genotypes of their parents? Well, let's write down the possible genotypes of these children. For A, this daughter could be big IA, or big IA, little I. For B, she could be big IB, or big IB, little I. For AB, she could only have one genotype. She has to be big IA and big IB. And remember that A and B alleles are codominant, and that's why she has both in her phenotype. Now for O, this daughter has to be little I, and that's because the little I is recessive to both big IA and big IB. Okay, now if you take a look, it's actually these two daughters that are going to tell us what the parental genotypes are. For the AB daughter, she had to get big IA from one parent and big IB from the other parent. So one parent has big IA and the other has big IB. To be O, she had to get this little I allele, one from one parent and the other from the other parent. So they both have a little I. So now we know the genotypes of the parents. Remember, even though there are multiple alleles for this gene, every individual can only have two alleles for the gene. So this one parent is big IA, little I, which means they're type A. And the other parent is big IB, little I, which means they're type B. And if you want to scroll back to the top, that means that the A and B daughters had to have been big IA, little I, and big IB, little I, because the other parent doesn't hold these big I alleles. So, to answer the question, the parents are big IAI and big IBI. Question 2. Emily is blood type A and marries Kevin, who is blood type B. If one parent of each individual had the O blood type, what is the probability that a child born to Emily and Kevin would have blood type B? So let's first look at their genotypes. Emily's blood type A, so we know she has to have at least one big IA. And we're told that one of her parents had O blood type, which means that the only allele they could give to Emily was little I. So this is Emily. Kevin is blood type B, so he must have at least one big IB, and one of his parents was blood type O, so again he must be little I there. Now if we're crossing two individuals, it says what is the probability that a child would be blood type B? So we could do a Punnett square. Big IA, little I, big IB, little I. So this child would be blood type AB, this child would be blood type B, this child would be blood type A, and this child would be blood type O. So the probability of blood type B is just 1/4, and it's this offspring here. Problem 3. In addition to ABO alleles for blood type, the Bombay gene H encodes for a substance that connects the A and B sugars to red blood cells. People with the genotype little h always exhibit an O blood type. For the following cross, what is the probability of an offspring with an O blood type? Well, let's see. How would you get an offspring with an O blood type? The offspring could be little I, with anything at the Bombay gene, and that would equal O. Or the offspring could have anything at the I gene and be little h, and this would also equal O. So these are what we're going to look for in the genotypes of the offspring. Now let's look at the cross. It's big IA, little I, big H, times big IB, little I, big H, little h. Now let's do the simplified line method that we've shown before. So when you're crossing big IA, little I, times big IB, little I, you have a 1:1:1:1 chance of all four blood types. So you have 1/4 big IA, big IB, 1/4 big IA, little I, 1/4 big IB, little I, and 1/4 little I, little I. Now for the H gene, we're really only interested in little h, little h. As long as you have one dominant H, that means that you will have sugars connected to your red blood cells. So as we did in previous problems, we can simplify the big H little h times big H little h to say that 3/4 of the time, the offspring will have at least one big H. And then 1/4 of the time, they'll be little h, little h. Okay? And you could just fill that in for all of these going down. Okay, now let's multiply across and see the genotypes. So let me just move this out of the way. Oops. So let me just move this out of the way. Okay, so we have 1/4 times 3/4 is 3/16 big IA, big IB, and at least one big H. And then 1/16 big IA, big IB, little h, little h. 3/16 big IA, little I, at least one big H. 1/16 big IA, little I, little h, little h. 3/16 big IB, little I, at least one big H. 1/16 big IB, little I, little h, little h. And finally 3/16 little I, at least one big H. And 1/16 little I, little h, little h. So now we're going to go through and see which one of these have blood type O. So what did we say before? Blood type O, you can be that by being little I, little I. So therefore, both of these individuals are going to be blood type O. And you can also be blood type O by being homozygous recessive at this Bombay gene. So that would be here, here, and here. So let's add these all up to see what percentage of the offspring is going to have blood type O. So you have 1/16 plus 1/16 plus 1/16 plus 1/16 plus 3/16. And that equals a total of 7/16. And that is the proportion of the offspring that will have blood type O. Question 4. Define and give an example of the following traits in humans. So sex-linked is when the gene is located on a sex chromosome. Now in humans, the traits that we normally talk about are usually X-linked, although the Y chromosome does have genes, so there could definitely be Y-linked genes. So we'll say X usually. And some examples of this, one is hemophilia, and another is red-green color blindness. And I'm sure there's many more examples for all parts of this, so if you have something else, you could just double check on the internet if it's sex-linked, limited, or influenced. Now for B, sex-limited means that its expression is in only one sex. So expression in only one sex. And some examples of this could be ovarian cancer risk, and that's because only females have ovaries. Or it could be prostate cancer risk, and that's because males only have prostates. Now for C, sex-influenced is when the location of the gene is irrelevant. However, the expression varies between the two genders, male and females. So expression varies between male and female. And two good examples of this are breast cancer risk, because it's certainly true that males can get breast cancer, it's just a lot more common in females. And another one is male pattern baldness, which you've learned about in the theory video. And that's where, in order for males to exhibit pattern baldness, they have to only be a heterozygote, but when to females have it, they have to be homozygous for this trait. So these are the difference between these three things. I think they're very easy to get confused, so if you want to just keep these definitions in mind, it could definitely help on a test.
Hi. Question 5. A study of midgetism in humans collected information from many families where both parents are heterozygotes for the midget gene. In every family, the grandparents were one midget and one person of normal height. You observe a ratio of 2 affected to 1 unaffected offspring. What does this data suggest? Well, first of all, before anyone asks, the term midget or midgetism may or may not be politically correct, but it is the technical term for certain forms of genetic dwarfism, so we're going to go with it here. Right off the bat, if you see a 2-to-1 ratio, almost always what's going to be represented? It's something to do with lethality or a lethal allele. If you're following along with the theory videos, we saw something like this in coat color in mice with the yellow gene. That's usually the example that's given. So that sort of catches your eye. It may or may not be true, of course, but if it was true, let's just look at what the lethality situation is. It generally means that if you cross 2 heterozygotes, and you would normally predict, of course, that you would get a 3-to-1 ratio of whatever trait is big M, which in this case would be midgetism. And why do I say that? Because 2 affected means 2 people exhibiting midgetism and 1 that are of normal height. So as you see in the cross here, you would really normally expect a 3-to-1 ratio if things were going normal, but so that implies that big M equals midgetism, little m equals normal height, and you'd have the 3-to-1 ratio, except in the lethal allele, this is the one, the homozygote for that dominant trait, that fails to develop, so it's lethal. So we see 2 in this case that are midgets, 1 of normal height, and that's exactly what you saw. Let's just make sure, though, that it goes along with the data. So what they said is both parents are heterozygous for the midget gene. Isn't that the cross that we just did? So if both parents are heterozygous, they didn't necessarily say it, but that implies also that both parents do exhibit midgetism, and that, we'll talk about the grandparents in a minute, you observe a 2-to-1 ratio. So that's totally consistent with 2 things. The fact that the midgetism allele is dominant, and that it's also homozygous lethal. If we want to just make sure the situation with the grandparents is okay, let's draw a little family tree over here, a pedigree analysis. The parents were both heterozygous, right? And we know that in their offspring, we saw some individuals, doesn't matter if they're boys or girls here. Let's just say there was a 2-to-1 ratio. 2 people affected with midgetism, which are also heterozygotes. 1 person who's not, and that's the 2-to-1 ratio, even though it wouldn't have to be exactly like that, right? Now the grandparents, they're saying in both of these cases, since both the cases are the same, we only really have to look at one of them, the grandparents were 1 midget and 1 normal height person. So we just have to ask ourselves, under our model, if it would be reasonable to get this individual from this type of pairing? Well, if you do the Punnett square for this one, it's a 1-to-1 ratio of midgets to normal height, but it doesn't matter because here we're seeing the 1, right? So, you know, it didn't say in every, well, it's funny, it did say in every case the grandparents were one midget and one person of normal height. They wouldn't necessarily have to be, but since that's the data, it's certainly consistent with what we said, okay? So again, they asked what the data suggests, and what it suggests very strongly. You know, notice they don't say prove because that's a much stronger word, but it suggests strongly that this form of midgetism is dominant and that it's homozygous lethal. Should we write that? Dominant and homozygous lethal. Okay, I'll see you soon.
Question 6. The dominance hierarchy for rabbit coat color is as follows, where full color, or agouti, is greater than chinchilla, which is dominant over Himalayan, which is dominant over albino. For A, a cross between a chinchilla rabbit, genotype c.ch, ch, and a Himalayan rabbit, genotype c.h, what is the predicted phenotypic ratio for the progeny? So let's fill out the Punnett square for this cross. You have a chinchilla rabbit, which is c.ch, ch, and a Himalayan, which is c.h, c. Okay, so if these are the genotypes, what are the phenotypes? Remember to always look back at your dominance hierarchy to get the phenotypes. So chinchilla c.ch is dominant over both the Himalayan allele and the albino allele. So for these two, they are chinchilla. So you have 1/2 chinchilla. The Himalayan allele, ch, is dominant over the albino allele. So both of these are Himalayan, so it's 1/2 Himalayan. Okay. Now for part B, you have another cross between a chinchilla and a Himalayan rabbit, but the genotype of the chinchilla is a little bit different. So the chinchilla is c, ch, and then c. And the Himalayan is ch, c. So when you fill out this genotype, this Punnett square, you'll see that it's a little bit different from part A. Okay, so again, because chinchilla is dominant over both Himalayan and albino, these two individuals are chinchilla. Himalayan is again dominant over the albino allele, so you have 1/4 Himalayan. And finally, this last individual is c, homozygous for this allele means that you're albino, so you have 1/4 albino. And this can be written differently as 2 chinchilla to 1 Himalayan to 1 albino. Question 7. Pattern baldness is a sex-influenced trait in humans. Males heterozygous or homozygous for the dominant allele, big B, little b, or big B, exhibit baldness, where females only exhibit baldness when homozygous dominant, big B only. A bald male heterozygous for the baldness gene mates with a non-bald female also heterozygous for the baldness gene. For A, what is the expected phenotypic ratio for each sex in the offspring generation? So I wrote 2 Punnett squares here so that we can just look at the B baldness allele and you could separate out the males and females. So in both, it's both male and female are heterozygous for the baldness gene. So it's going to be big B, little b, big B, little b. And as you can see, that's a simple monohybrid cross that we were doing in the first Mendelian chapter. Okay, so let's write this one can be the male Punnett square, male offspring, and this could be female. So because it's sex influenced, that means that it has a different phenotype in each sex. So for males, in order for a male to be bald, he only has to be heterozygous for the big B allele, or he could be homozygous for the big B allele. So if these are the males, which males in this offspring are bald? You have big B, homozygous dominant. That male is definitely will exhibit pattern baldness. However, these heterozygous males will also exhibit this condition. So for the males, you have 3 bald to 1 non-bald. And remember that these are males. Now for the females, females only exhibit pattern baldness if they're homozygous dominant for this condition. So that means that only this female here is going to exhibit pattern baldness. So for the females, you have 1 bald to 3 non-bald. And these are for females. Okay, so these are the phenotypes. Now for part B, they're saying what are the expected offspring genotypes and in what ratios. So now we're going to have to look at both genes, both the gene for baldness and also the sex genes. So let's see. For B, let's write the parental cross again. Again, the father exhibits baldness but is heterozygous, so big B, little b, and because he's male, he's XY. Now the mother is heterozygous but does not exhibit pattern baldness. She's big B, little b, and she's female, so she's XX. So now let's do the simple line method to look at the genotypes for both. So if you cross big B, little b times big B, little b, again from above you have 1/4 big B, 1/2 big B, little b, and 1/4 little b, little b. Now when you're crossing XY times XX, you have a half chance of being XY and a half chance of being XX. So you're going to draw that here. 1/2 XY, 1/2 XX. And the same for all of the other combinations of B. Okay, now to find the genotypes and their ratios, let's multiply across. So you have 1/4 times 1/2, and that's 1/8, big B, XY, and 1/8, big B, XX. Now you have 1/2 times 1/2, that's 1/4, big B, little b, XY, and 1/4, big B, little b, XX. Another 1/4 times 1/2 is 1/8, little b, XY, and 1/8, little b, XX. So these are the phenotypes, the genotypes and the ratios, which is what the problem asked for. But why don't we just look at phenotypes just to double check. So again, these are the genotypes. Now let's look at the phenotypes. If you're big B, XY, you're definitely male, and you definitely will exhibit this pattern baldness condition. So we'll say male and bald. If you're big B, and XX, you're female. And because you're homozygous dominant for this baldness allele, then you're going to be bald. Okay? For big B, little b, XY, you're male. And again, in male, heterozygotes do exhibit pattern baldness, so you would be bald. And big B, little b, XX, you would be female and non-bald, and that's because, again, this is a sex-limited trait. And so it's different, has different phenotypes in each sex. Okay? So to be little b, XY, you're definitely male, and you're non-bald, because you don't have any allele for the baldness trait. And 1/8 little b, XX, you're female, and you're non-bald. Okay, so if you want to add all these up, let me just write the numbers next to here. So 1/4 again is just like 2/8, so it'll be easier to add. Okay, so let's say male, female. For male bald, you have 1/8 plus 2/8, so that's 3/8 bald and 1/8 non-bald. And for females, you have 1/8 bald and then 2/8 plus 1/8 non-bald, so that's 3/8 non-bald. So that definitely corresponds with part A and shows that we did do it right. So it's 3-to-1 bald to non-bald in males and 1-to-3 bald to non-bald in females. So that answers the question on the sex influence trait in humans.
Let's look at the true-false section. All of these problems have something to do with dominance relationships and or epistasis or something like that, since that's what this whole section is about. So let's check it out. Number 1, a person with blood type AB can donate blood to individuals with blood types A, B, or O. Well, this is obviously testing your understanding not of the, there's not a cross going on here, but just about understanding compatibility of transfusions between people with different blood types. AB basically is the universal receiver, but AB individuals can only donate blood to other ABs. The reason is because the antigens on the B antigens and the A antigens on red blood cells will cause a reaction in any of these other types. So O negative, you may know, or O is generally the universal donor, but the way this question is phrased, it's definitely false. A woman, number 2, with blood type A and a man with blood type B could produce a child with blood type O. Well, it's true, they could, but how could they? Let's do a little crossover here. The woman would have to be capital IA, little I. She'd have type A blood, right? The man would have to be capital IB, little I, and then there'd be 1/4 probability that the two little i's would come together and make someone with type O blood. So they didn't really say how frequent it was, they just said could it happen, and absolutely yes. Epistasis is an example of multifactorial inheritance. That's true because multifactorial simply means more than one factor, and what it really means is it's not as simple as what Mendel originally envisioned, that you have just one gene with, 2 alleles. Usually it implies either that there are more than one gene pair, which is what, I shouldn't say it's what epistasis is, but epistasis is an example of that, so we just answered our question. Epistasis is defined as the interference of one gene's action on another. So that's one example of multifactorial inheritance. Also, the environment could be involved. There could be 20 genes, but this is definitely true. Now, number 4, a heterozygote with codominant alleles would have a phenotype somewhere in between the homozygous phenotypes for the 2 alleles. What that's describing is what? It's not codominance, it's incomplete dominance. If it was codominance, we have said that the phenotype of the heterozygote would be to express both of the traits present in the homozygotes, right? Like in the case of AB blood, you're both A and B at the same time. There's no somewhere in between there. So that is definitely false. 5. Epistasis can be defined as 2 non-allelic genes affecting one trait. I'm going to say true for that one with one little star there. Technically, epistasis is defined as interference of a non-allelic gene on the action of another non-allelic gene. The situation with rooster comb type, for example, technically is not epistasis to me, but there are some textbooks that might imply that it is, because those are two non-allelic genes that are contributing to a trait equally. So it's going to depend. This gets very, very picky, I guess you would say, technical. Some people use epistasis in this more general sense. Some people are a little more specific about the interference effect. Number 6, pleiotropy is defined as one gene product having effects on multiple traits and characteristics. And that's true. Why? Because that is what pleiotropy means. I know that isn't very helpful. Well, I can tell you this. Pleio means multiple or many. So pleiotropy sort of means many traits. An example of this, it gets a little hard sometimes, but sickle cell anemia is one gene. Now, the problem is in hemoglobin, but before they really understood this, they said, look at these individuals have all sorts of seemingly unrelated things wrong with them. So they said it was a pleiotropic trait. 7, the expressivity of a gene can depend on an individual's genotype, age, gender, and environment. That's definitely true. Expressivity means the degree to which you are exhibiting a trait that you genetically have. So the best example I can think of is Down syndrome, just as an example. There's definitely variable expressivity about this. Some people with Down syndrome are very low functioning and some are extremely high functioning. Now, why exactly that is, it's very often hard to say, but clearly all of these factors might play a role, right? We don't know. Certainly the environment is a very broad term. So expressivity is what it is. Why it happens cannot always be determined, but certainly these are the type of factors that we suggest are responsible, or at least contributory. Number 8, response mechanism of genes to sex hormones is not used to explain sex-limited inheritance. Now, sex-limited inheritance means certain traits only show up in one sex. So what would be the explanation for that? It's exactly what it says here. Sex hormones and your response to them. If there's testosterone around, the body responds in a certain way. It expresses certain genes that are different than if there's estrogen around. So the key here is if you just took the not out, response mechanism of genes to sex hormones is used to explain it, it would be true. Since they're saying it's not, it would be false. This can get, obviously, guys, true, false things, they're not the most difficult thing in the world. But when they start throwing nots or multiple nots or tell me which one is not true sometimes, you know what I'm saying. Just be careful. And lastly, overdominance occurs when heterozygotes have superior traits. Well, that's true, and again, it's not very helpful, but that's the definition of overdominance. So sometimes this phenomenon is referred to as heterozygote superiority or hybrid vigor, you may have heard. But a great example is with the sickle cell anemia gene. It's recessive, but heterozygotes are better off evolutionarily than either of the homozygotes. Certainly if you have sickle cell anemia, that's not great. But it turns out that individuals that are big S have a susceptibility to malaria that these individuals don't have. So heterozygotes survive the best. And that's what, it is true, but that's what overdominance is, hybrid vigor, heterozygote superiority, and there's probably even another trait for that I can't think, another term for that I can't think of. All right, guys, great job so far. We'll see you again real soon.